My name is Muhammad Al-Tanani. I'm a professor of molecular pathology and cancer therapeutics in University of Bradford, based in Institute of Cancer Therapeutics here. I appointed here as an university chair, which is called in United States as a distinguished professor in molecular pathology and cancer therapeutics. Before I appointed here in University of Bradford, I was in a medical school in Queen University, Belfast in Northern Ireland, and I moved recently here from about 3 years in Bradford. I moved with me my research laboratory to carry on what some of the things I did in Queen University, Belfast. My research laboratory focused mainly about breast cancer and breast cancer metastasis. That means the primary tumor, and when this primary tumor started to detach from the primary site and moving through the circulation and the lymph node and homing in other organ, like it's moved from breast to lung or to the brain, what is called the metastasis. My research has two arms, is to identify a new therapy for breast cancer and also a novel diagnostic for unmet breast cancer. Breast cancer is not one disease, it's several diseases. That's why I said a novel diagnostic for unmet subtype of breast cancer. I focused in last maybe 10 to 15 years in one subtype of breast cancer. It's called the triple-negative breast cancer. And the triple-negative breast cancer is there are three genes not expressed in the tumor of this patient. It's estrogen receptor negative, progesterone receptor negative, and also is HER2 negative. And that's the treatment of this patient become very limited in a way. There is no target therapy for this patient like anti-estrogen or the HER2 therapy, Herceptin. And first line of treatment of this patient is a chemotherapy. It's well known as a chemotherapy has a side effect, and is major side effect in the normal cell, normal organ, is eventually is a poison, which is poisons the cancer cell and also the normal cell. Therefore, the best therapy for cancer patient in general, what is called the targeted therapy. That's a drug which is specific to a switch of certain target genes. Therefore, I focus myself and my research laboratory in Queens, as well in Bradford, to identify a drug which can inhibit a specific target or targets, and then that it will be a good option for the patient before, instead to go for chemotherapy, to maybe stay for 10 years, 20 years in targeted therapy. I moved to identify the gene or a set of genes which involved and contributed in malignant transformation and the metastasis of triple-negative. It takes a long time from my research group. We started in 2006, and we used advanced technology at the time to identify the genes which involved in initiation and development of triple-negative breast cancer. From this technology, we end up by a very long list of target genes which might be contributed. And then we did a lot of work in the laboratory in vitro, that's in petri dishes, and also in vivo in animal model system, to identify which one or which set of genes very important and most abundant and most effective in initiation and on development of triple-negative breast cancer. From these genes, we went down from hundreds of genes to one single gene, and this gene is called RAN-GTP. And RAN-GTP has a very important physiological role in our normal cells. Do a lot of work with cell cycle progression, as also with nucleocytoplasmic transport. They take the cargo from the cytoplasm to the nucleus, and any cargo or any protein or come from the cytoplasm to the nucleus is need RAN-GTP to the associated from the inhibitory complex to be used in the nucleus after that, and also is involved in mitosis and meiosis. That has very important physiological function. At the time, we didn't understand how these genes involved in cancer and cancer metastasis. But to do that and to validate that, what we do, we used advanced genetic engineering model system to switch on and to switch off. And we got a very aggressive triple-negative breast cancer cells in our lab. And by genetic engineering, we switch off RAN-GTP and study if we switch off, we affect the cancer cell. And what we found, when we switch off, we killed about 19 to 95 of triple-negative breast cancer in vitro, in petri dishes. But because RAN-GTP has very important function in our normal cell, we need to make sure when we switch it off has minimum effect or no effect in normal cell. And what we did really, we switched off to sometime between 50% to 90%. That means remaining only about 10% is active and expressed of this protein. And we find the cells function very well. The cells do not die, the normal cell. But when we switch it off from cancer cell, the cancer cell vanished. And by this genetic engineer thing, that's very good news. But we said, okay, we switch it off in highly aggressive breast cancer. What will happen if I switch it on in a normal or near normal breast cell or in a benign non-malignant breast cells? We switch it on, and we found when we switch on, the phenotype of the cells changed from near normal to become malignant and also become very aggressive and invasive in other organs. And we shown that in vitro and also in vivo in animal model system. The same thing is happening in benign. It changed the benign to become malignant and highly invasive. And therefore we said, it can be if we got a drug which can switch off this gene by 90% or 95%, that might be we're going to mimic what we found by genetic engineering. And we used a very advanced, complicated methods to identify the drugs which can switch off this gene. And we got a very long list of drugs. Potentially can inhibit the RAN-GTP. And in the top of this is a drug called the Pimozide, and Pimozide is anti-psychotic. Therefore, we come to this paper which will publish in Oncotarget, which the title of the paper is Anti-psychotic drug Pimozide is a novel chemotherapeutic for breast cancer patient, mainly triple-negative breast cancer patient. For this work, my laboratory collaborated with high-profile other scientists in different universities. From them, found a long list of authors. Professor Philip Rodland in University of Liverpool, Professor Richard Morgan in our Cancer Institute here in Bradford, Dr. Gross in Nottingham, and other people really contributed to this work with my research group. And then we tested Pimozide. We started to say, okay, we need to test Pimozide. We tested all of them including the Pimozide. But Pimozide come in the top of the list for our selection for the reason I come now to tell you about it. First, we found when we treated a very aggressive, a big panel of breast cancer cells, we found Pimozide induce what's called the cell death program. That's mean the cells go for death, and eventually Pimozide kills the triple-negative breast cancers and other type of cancer, including lung cancer. And I'm going to tell you why we choose lung cancer to test it, if Pimozide can also kill the lung cancer. And one of them is very resistant to a lot of chemotherapies called the non-small cell lung carcinoma, lung cancer. And what we found is Pimozide, when we treated the cells, aggressive cancer cells, with Pimozide, kills the cancer cells in vitro in our laboratory. And then we said, okay, we need to see if we're going to mimic the same effect of genetic engineering we did in a normal cell. When we switch off in a normal cell, we not by genetic engineering, we not found any effect in the normal cells. That means the normal cell still grow fine, still function fine, and all of the other thing. We need to make sure also is if we're going to do more research, a more deep research about Pimozide, is a Pimozide not kill the normal cell, because in the end of the day, when we come to treat the patient, we need to kill only the cancer cell, and we need to keep the normal cell fine and intact, and all the organ working very well, and so on. Therefore, when we treated a normal cell, different to normal cell, by Pimozide, we found very minimum effect, if there is any effect. And this is encouraging us to say that is the drug which we're going to spend good time and effort to find out if this drug can be useful for our patient. And then after that, we used animal model system. We got the triple-negative tumor implanted in the animal, and then we treated the animal with the Pimozide. The surprise in our result and our data, and this is published in Oncotarget, we identified is a drug, the Pimozide, is not only shrunk the tumor by 60 to 70%, which is very good. The primary tumor shrunk it by 70%. That's a great result. But not only that, but we found that Pimozide nearly prevent these cells to detach it from the primary site and invade other organ. That means is prevent the cancer metastasis. And this really was very encouraging to go deep and understand how when you switch NGTP by Pimozide, you kill the cancer cells, the primary tumor, and prevent it to invade other organ like lung or brain or whatever, or liver. We did a lot of study in vivo and in vitro to understand how the drugs work. Why first shrunk the tumor, and also how also the drug is prevent the primary tumor to invade other organ. How this happened to suppress all of this? It should be there is a mechanism for that, and it's very important for us to understand how the drugs work. Yes, it's good news, it's victory if we found is shrinking the tumor prevent, but we need to understand. And what we found is Pimozide can inhibit one of the major hyperactive pathway in triple-negative breast cancer. It's called the PI3K AKT mTOR pathway or signaling pathway. We found inhibit AKT, and inhibited the phosphorylation of this protein. And this is one of the hyperactive pathway in triple-negative breast cancer. But is again how shrinking the tumor, yes, this inhibit this signaling pathway control hundreds of other genes. And also RAN-GTP in its own is control and interact with other maybe 80 to 100 different genes. That's mean when you switch off RAN-GTP by Pimozide, you not only switch RAN-GTP, but you switch other 100 genes and pathways underneath RAN-GTP. We found some of very important genes in cancer development downregulated, that's mean inhibited. For example, we found hepatocyte growth factor receptor is downregulated. It's not only that, we need to understand also more deep how we shrunk the tumor first, and then after that we need to understand how is prevented the metastasis. How shrunk the tumor? We found Pimozide is affect the what's called the angiogenesis, the vascularization of the tumor, is inhibit the main receptor which can involve it in blood supply for the tumor, is nearly stop the blood supply when you to the tumor when you treated the tumor by Pimozide or when treated the cancer by the Pimozide. And this is called the VEGFR1 and the VEGFR2, vascular epidermal growth factor receptor 1 and 2, found when we treated the animal by Pimozide. These two main receptors for angiogenesis downregulated. And therefore we understand now in somehow how this shrinking the tumor, first by downregulated a major hyperactive pathway, PI3K AKTM2 pathway. The second is by inhibiting the angiogenesis target genes, VEGFR1 and the VEGFR2. And the third of these we found is also is reduce the fibroblast, which is involved in tumorigenesis and metastasis. But that's a part of understanding how Pimozide is work. But it was a part from the thing. The other thing we found when we treated the animal by Pimozide, in the tumor is become the lead to what's called DNA damage. That means is it change even the DNA and lead to the cells to go for apoptosis. The other thing is very important is how Pimozide prevent the cells to metastasize, to move from primary site to go to the secondary site. And this is really very hard and very difficult to find one drugs which can shrink the tumor and also inhibit metastasis. Have two big rules here. But it should be, there is a mechanism for that, and we have to understand how this happened. And what we did really is two things. We found, or several things to be honest, we found is one of the target gene inhibited by Pimozide is called the matrix metalloproteinases, is involved in metastasis heavily and well known in our scientific culture is MMP is the main target to dissolve the connective tissue and make the cells to go to penetrate penetrate the vascular system and go through our circulation and invade other organ. And we found Pimozide inhibit a group of metalloproteinases. It's not only one, it's a group of metalloproteinases, of matrix metalloproteinases. The other thing we found is very important, is inhibited other signal and transduction. It's called EMT, is epithelial-mesenchymal transition. We found is when you treated the animal or the cells or the cancer cells by Pimozide, is EMT transcription factor go down. And in other group, they identified that Pimozide can kill what is called cancer stem cell. And the cancer stem cell, in brief and for in general way, is very tiny number of cancer cell within the tumor responsible about the metastasis, which this group of cells is very a small number of cells within all the tumor. You found only small number of cells is called the cancer stem cell, which is resistant to most of chemotherapy and drugs. And this cells, which is mainly involved in tumor initiation and also in cancer metastasis. And the other group, they identified that Pimozide can kill the cancer stem cell. And when we did the work, we like really to prove that because, and to identify that if this is happening in our model system in triple negative breast cancer or not, we tested the EMT transcription factor, and we found is Pimozide, which is a RAN inhibitor, is inhibited all the EMT transcription factors. And then and each factor under each one is other target genes. And this target EMT, when it's switch on, is it change even the some of the tumor cells, which is not cancer stem cells, to be cancer stem cell. And involve a lot of signal transduction under EMT and above EMT when it's switched on. And we identified Pimozide is inhibited EMT. That means is kill the cancer stem cells. And this is a rationale, several rationale, how Pimozide is not only shrinking the tumor to nearly 70%, but prevent this tumor to become aggressive, and then after that is detached from primary site, moving through circulation, lymph node, and homing in other organ like lung. And we identified the animal which is treated by Pimozide, their lung is clear, like exactly the lung of normal animal has no cancer. In compare with the animal is not treated, we found that tumor moved from the breast, the primary site in the animal, to the lung, and the lung morphology and histopathology is changed in untreated animal. But in treated animal, the lung very close to the normal animal. That's why really it's very important now to find out the mechanism. We found the mechanism is angiogenesis and the cancer stem cell. And from this result, we identified how this works. But the next step we thought about it is any relation in the tumor between RAN and also the VEGFR in the tumor, in human tumor. That means is any association between the two genes. When RAN GTP go up, is VEGFR1 or 2 go up as well. When they become down, it's become down, and this is involved in our association in patient survival. And what we found, when RAN go up, the patient survival go down, and in the same chamber, the same tumor, we found VEGFR1 and the VEGFR2 go up as well, and the survival of the patient go down. That means a RAN GTP is controls the VEGFR1 and 2 when switched on RAN GTP, VEGFR1, which is and 2, which are responsible about angiogenesis, new formation of vasculars around the tumor to make the blood supply and make the tumor more grow is increased when RAN GTP increased. That means RAN GTP is when you switch it on, VEGFR1 and 2 switch it on, and the blood supply for the tumor increase and therefore become more aggressive and metastasize. What we found in a patient which is the survival rate is very high, we found RAN-GTP switched off and VEGFR1 and 2 switched off, and this is the survival rate of this patient very high. This is a study already in this paper, and you can read it. What's the next step now? This is a drug which can shrink the tumor and also prevent the tumor to metastasize. What's the next step? The next step for us, for ourselves now, we're seeking to take this drug to clinical trial. Because this drug is already in clinic, the patient with schizophrenia treated by this drug, that's mean all pharmacological study, pharmacokinetic study, and toxicological study has been done, which this is a clinical trial phase 1. We do all the pharmacokinetic thing. That's mean now, if we have the money to do the clinical trial, we don't need to go for phase 1. We can go directly to clinical trial phase 2, or we can combine what's called the phase 1, phase 2 together. We don't need a phase 1, a proper clinical trial phase 1. And if we go for clinical trial phase 2 by Pimozide, that it will be the first targeted therapy for triple-negative patient for about 97% of triple-negative, because triple-negative has only 3% of them have a targeted therapy if they have a specific gene is not function. It's called the BRCA1 or BRCA1. If BRCA1 mutated in triple-negative patients, they can treat it by PARP inhibitor, and this is only 3% of all triple-negative breast cancer. 97% of breast cancer patients haven't targeted therapy and depend only about chemotherapy. If Pimozide or RAN inhibitor, in a specific RAN inhibitor, being in clinic, that means it will be the first targeted therapy for triple-negative. That's my hope, and I wish really to take this study to clinical trial, and will be in clinic as soon as possible to help our people, our patient, for a targeted therapy which can be for treated by tablet for 10 years or 20 years, so it will be like a chronic disease. Thank you.
Oncotarget was launched in 2010 as a traditional peer-reviewed journal with free access that focused primarily on cancer research. Currently, Oncotarget is a multidisciplinary journal which publishes in weekly issues and is covering topics beyond oncology, in fields such as neuroscience, immunology, pathology, and more. You can click on the link in the description below to order a reprint or read the manuscript that was discussed in this interview on Oncotarget.com. Please feel free to subscribe to our YouTube channel and connect with us on Facebook, Twitter, or LinkedIn. Thank you.